Well, welcome to Doing DNA, the Code of Life. I'm Tom Zinnan. I'm here at the UW-Madison Biotechnology Center with my colleague, Liz Jesse. Liz.
Hi, I'm Liz Jesse. I am at home, not at the Biotechnology Center. So you'll get to have a little bit in the Biotech Center and a little bit from home, which is probably where most of you are.
So we're going to talk today a little bit about DNA. Here are replicas of 3 Nobel Prizes in medicine or physiology awarded to researchers at UW-Madison for work done on DNA. There's actually a 4th one that's been awarded since this was made back in the late 70s. So this is a pretty good place. It's a DNA university. Some fantastic discoveries about this thing here, which is our little model of DNA. We're going to say DNA is a lot like a recipe card because and first we're going to say DNA is a lot like an alphabet. So here's one model of DNA, and here's our twisted helix, double helix fountain. And it is the oldest and most accurate DNA fountain in the world. It's not plugged in right now because of COVID, but I want to point out to you that there's 2 strands, this strand and this strand, and there are 4 letters. Now DNA is a lot like an alphabet. In the English alphabet, there's 26 letters, A through Z, and you can tell the different letters because they have different shapes. Likewise, DNA is a lot like an alphabet, but it has only 4 letters, and we tell them apart by their different shapes. Here's the first letter. It's a 9-sided one with 2 pipes sticking out of it, and that one is almost always opposite the 6-sided one with 2 pipes sticking out of it, and vice versa. And that is half of the DNA alphabet right there. If you've heard of A and T and G and C, this is A, and that is T. Here's the second half of the DNA alphabet. Here's a 9-sided letter with 3 pipes sticking out of it. It's almost always opposite the 6-sided one with 3 pipes sticking out of it, and vice versa. So this is G and C. And what's cool about this is just like with English, the letters in sequence carry information. And if you change the order or the spelling of the letters, you can change the meaning of the words. And that's a big deal. So with this, for example, I'm going to tell you a bad joke here. You can tell that this is a boy DNA because it is a helix. If it were a girl DNA, it would be a shelix. So with the addition of 1 letter, you can make a bad joke, but you can also make a good point, and that is small changes in the order or sequence of the letters, whether it's in DNA or in English, can make a big difference in meaning. I'd like you to look at this one because this is a pretty good metaphor for what we do in science. Please look at this one and tell me, are these 2 yellow screws identical? They look like they are, but are they really? Let's test that because we are scientists. If you put a car here and you put a car here, and if you're driving this car and if you're driving this car and you go like this, which way do you have to turn the steering wheel for this one? And which way do you have to turn the steering wheel for this one? So while they look identical, they're actually distinctly different. They're mirror images. And you can do that with your hands. If you say, please raise your right hand, and then please raise your other right hand. I can't do this because, well, maybe I'll be able to do it this way. Oh, yeah. Please raise your right hand, and then please raise your other right hand, the one that is left. Put them together and ask yourselves, are they identical or are they symmetrical? Are they mirror images? And this is a big deal. This is a great metaphor for one of the functions of education at any level, and that is to help train your brain to be able to learn to discern between 2 things that are very similar yet distinctly different. So now we're going to go down the DNA hallway. Here's our little racetrack that gets across handedness. Here's our 1 model of DNA, double-stranded, anti-parallel, helical, and complementary. And we'll grab this guy and take it with us. Now, when you come to the BioTrek Center, we could take you on a tour down our DNA hallway. And this installation of art was done by a professor and assistant, an artist here. And if I ask you, how do the Canadians spell harbor? You may know that the Canadians spell harbor H-A-R-B-O-U-R. And if I ask you, how do the Canadians spell color? You know, the Canadians spell color C-O-L-O-U-R. And so they put an extra letter in there that Americans don't use. And sometimes, if it's even a little bit different, how do the Canadians spell center? C-E-N-T-R-E. And so you can have variants in the same general language, but slight differences in how they're spelling. In those cases, it doesn't change the meaning. But likewise with here, we're going to have the same word as it is spelled in DNA, with the 4 letters of DNA, in 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 different species. And I'm going to try to show this. Liz, if you can let me know if this is popping up or not. I don't think it is. The words, it's the same general word, but you can tell that it's not the same length of spelling, because here's how it ends in this bacterium called E. coli, and we're going to go down and look at all the other species. So there it is with E. coli, and here it is with this, which is a bacterium. Here it is with this roundworm or nematode called C. elegans. And here's the word with Homo sapiens, which is humans. And this is where it ends with humans. And you might think that's the longest one, but watch what happens. Here it is with a toad or a frog, Xenopus levis. Here it is with zebrafish. And the word is the longest here. It's even longer than it is with humans. That's the vertebrate. Here's the fruit fly. These are all examples of model organisms. Here it is with corn, Arabidopsis, a little weed, Aspergillus rhizae, filamentous fungus, and Saccharomyces cerevisiae, the brewer's yeast. And so this helps give you an idea that DNA is a lot like an alphabet, and it's also kind of like how we spell words in English. There can be different spellings or variants of the same word depending on what species you're in. And this is important because it also illustrates, again, model organisms. Model organisms have 4 big characteristics to them, from my point of view at least. They need to be small, fast, simple, and cheap. Small, fast, simple, and cheap. And that makes them really good for doing fast work in genetics. You could ask yourself, if you're a plant geneticist, do you want to work on redwoods or radishes? And it's great to work on redwoods, but radishes are small, fast, simple, and cheap. And so you might want to think about doing your genetics work with something like Arabidopsis because of those great characteristics. DNA stands for deoxyribonucleic acid. What does that mean? Well, it's the acid thing originally found in the nucleus. It's got this 5-carbon sugar called ribose, and it's missing an oxygen. So that's all it means. The name was assigned before the function was known, before we knew what it did. So it's the acid thing found in the nucleus that contains this sugar, ribose. It's got 5 carbons and it's missing an oxygen. You might know or heard of RNA, which is its cousin, ribonucleic acid. And that's pretty similar to DNA. All right, now we're going to go into the lab. So here we are in room 1340. This is where you would come if we were going to be doing a field trip with your group. And what's cool here is in this teaching lab, they use fruit flies. And here are some adult fruit flies. They go, Liz, how quickly can they go through their life cycle?
About 10 days.
Yeah. And so you can get about 25, 26 generations a year. And so this gives you an idea of why it is that you would like to be able to use model organisms. In general, they're small, fast, simple, and cheap.